Pathogenesis. How HLA-B27 initiates AS is unknown, and, after many years, some of the earliest hypotheses are still being investigated. The original hypothesis, called the arthritogenic peptide theory, suggests that the presentation of either bacterial peptides by HLA-B27 or self-mimicking HLA-B27 binding peptides from certain bacteria could initiate a cell-mediated immune reaction leading to AS. The second one is the unfolded protein response hypothesis, which suggests that HLA-B27 tends to misfold and accumulate in the endoplasmic reticulum, triggering a stress response that results in the release of IL-23. However, a new study has questioned these two theories, claiming that the arthritogenic peptide theory should be reassessed in terms of quantitative changes concerning self-peptide presentation and T-cell selection. It also stated that the absolute binding preferences of HLA-B27 allotypes are not sufficient to explain the association of the disease. The third hypothesis is the HLA-B27 homodimer model, which supports the view that HLA-B27 homodimers have an abnormal interaction with natural killer, NK, and CD4 T cells. Unlike the heterodimeric form of HLA-B27, the homodimer is able to bind to certain killer cell immunoglobulin-like receptors, KIRs, which are expressed on NK cells and T cells, causing the release of IL-17. Ridley et al. Have proven that CD41 T cells upregulate the expression of KIR3DL2 on the cell surface and that the binding of this receptor to HLA-B27 potentiates T cell survival and Th17 cell differentiation. Th17 cells are a type of T helper lymphocyte which produces IL-17, a cytokine able to increase T-cell priming and stimulate immune cells such as fibroblasts and macrophages, promoting the release of IL-6, TNF-alpha, and other chemokines. Oppmann et al. Found that IL-23 is one of the triggers of the Th17 response. This molecule is a pro-inflammatory cytokine that seems to play an important part in stabilizing Th17 cell phenotype through the transcription factor BLIMP1, PRDM1, which is associated with Crohn's disease. Th17 cells are commonly found in the intestinal lamina propria of the gut, and their instability can be induced by exposure to certain bacteria, leading to the transition of Th17 cells into regulatory Treg cells. Maxwell et al. Also suggested that IL-23 promotes the accumulation of Treg cells in the bowel, some of which probably were Th17 previously.